Okay, so next chapter, as we mentioned before, we just finished the lipid part. Next, we will move to the protein part. Depends on, as I mentioned before, depends on the way they incorporate into the membrane. There will be the three classes of the membrane protein: integrate membrane protein right here, or transmembrane protein. They will penetrate entirely across the hydrophobic core. And the lipid anchor protein, they will use kind of conjugate with certain lipid and use those lipids. There will be certain lipid conjugate with the amino acid. It's kind of modified protein, and they use those lipid to integrate into the lipid. And the proliferate membrane protein here, they will bind to the other lipid, so they indirectly associate with the membrane. They call the proliferate membrane protein. They all need to deal with such a hydrophobic property. So for the integrate membrane protein, as you can see here, this is really typical integrate protein or transmembrane protein. You will see this is a hydrophobic core, right? So they will arrange their hydrophobic or non-polar amino acid in this part and arrange their polar or hydrophilic region in this part. So this will be the N-terminal in this case, will be hydrophilic or polar amino acid, then become alpha helix, which usually are hydrophobic part, then hydrophilic, and then make a turn, go back to the proximate hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, like this. So if you check their kind of amino acid sequence, you will see that it's always kind of have a kind of, there's a polar amino acid, non-polar, polar, non-polar, polar, non-polar. So this red part, non-polar region, we call that they will be kind of more likely assemble or integrate into the membrane part. Okay, we call this a transmembrane domain. Okay, and those are transmembrane domain usually contain about the 20 to 25 amino acids, as you can see here. Okay, this is alpha helix, which typical the secondary structure of the transmembrane domains. Green and the yellows, they are hydrophobic amino acid. Okay, most of them will compose of the hydrophobic amino acid. That is why they will be kind of stable here. Okay, and it depends on, so you can calculate the percentage of the hydrophobic amino acid in certain area and get the so-called hydrophobicity index through the easy kind of computer website. So it will give you such an index. So with a higher index, it means this area is most likely will be the transmembrane domain. And with a lower hydrophobicity, they will be the hydrosoluble. They will be the kind of hydrophilic amino acid. So they have no chance to be kind of transmembrane domain. So based on this, we will kind of predict glycopurine only have one transmembrane domain. But based on this, you will see the many different kind of hydrophobic area with a high hydrophobic index. So they are multiple. This protein contain multiple transmembrane domain. Okay. So this is an example. Glycopurine A, which is only, this is glycopurine A, it's only contain one transmembrane domain here. Okay. And this will be the example for the multiple transmembrane domain protein, like bacterial rhodopsin, glycerol channel, or these two. And in some case, they will use the one continuous alpha helix to act as a transmembrane domain. But in some case, it's not so easy in some cases to predict the number of transmembrane domain because they're somehow really tricky. For example, here, they use this soluble part here, then form half of the transmembrane domain, then go to the other side, then go back here and form the other half of the transmembrane domain. So if you check this, you don't see that the typical length of the transmembrane domain. You will kind of really easy to kind of skip that or ignore that part. Okay, so it still need, in some cases, still need kind of 3D protein structure. Now we have AlphaFold, so it will be much easier to predict this kind of solve this question. Okay, and there will be few kind of example. For example, this will be the example for the other multiple transmembrane domain protein. It's aquaporin. Aquaporin, the people found the aquaporin got the Nobel Prize. Okay, because that's a kind of water channel. They kind of can provide a channel for water to go across the plasma membrane. You will see here, this is a cartoon dynamic simulation. The red will be the water molecule. This is cytosol part. This is extracellular part. This is the plasma membrane. And this yellow will be the polar head of the plasma membrane. And the green will be the fatty acid tail of kind of plasma membrane. The blue one will be the alpha helix of the aquaporin. And inside the aquaporin, it will be a lot of kind of, in the aquaporin, it will be kind of water tunnel, okay, which should be the hydrophilic. The water will go through that water tunnel or channel across the membrane, okay? But how such a hydrophilic property can make aquaporin embedded in the plasma membrane? The water tunnel is inside the interior of the aquaporin. The surface of the aquaporin is still hydrophobic. This is the surface, the white one will be the surface of the aquaporin. They are super hydrophobic. They are still alpha helix. Okay, there's only one hydrophilic tunnel inside that a lot of multiple alpha helix. Okay, so in the surface it's still hydrophobic. That is why they can easily kind of provide hydrophobic interface with the plasma membrane. They'll be easy, kind of, or stable their position there. Okay, but inside will be the hydrophilic. Surface will be the hydrophobic. Okay. So typically, transmembrane protein will be built by the non-polar amino acid and the alpha helix, but there will be always exception in the biology. For example, this will be kind of several transmembrane protein in the T cell. T cell is kind of one kind of immune cell. Once they kind of be activated, they will trigger the downstream immune response. But to trigger the T cell signaling, several signaling molecules in the ER of the T cell, they need to assemble, form the cluster like this. So usually those transmembrane protein, they only can do the electrodiffuse, right? And they, of course, they still use alpha helix as their kind of transmembrane domain, but not all amino acids are non-polar amino acids. Part of them are polar amino acids and have charge. So those electrostatic force can help them during the electrodiffuse once they need to be activated. Those electrostatic force can help them to assemble each others and finally form the stable cluster to trigger the downstream immune response. Okay, so that would be the one exception. And also not all transmembrane domain are alpha helix. The other kind of typical secondary structure is beta strand, right? Beta strand usually considered as a hydrophilic secondary structure. Alpha helix usually considered as hydrophobic secondary structure. But here, it's kind of, this protein should be the porin, okay? Porin is also the transmembrane domain, but they use beta strand as their transmembrane domain. But again, they still arrange several hydrophobic protein on the surface of their beta strand, shown in the yellow color. So they still make their surface super hydrophobic. But inside this barrier will be the super hydrophilic. But they don't care because there's no lipid inside. They always kind of need to, they only need to kind of generate hydrophobic interface then kind of to interact with a membrane that can make them super stable layer, okay? Okay, that will be the transmembrane domain protein. And second, next will be the lipid anchor protein. So as you can see here, those blue dots, they are each kind of many different amino acids. So those are protein through several kind of enzyme reaction, they can conjugate different lipid onto the polypeptide. So there will be different modification here. For example, acetylation, prenylation, and the GPI anchor. They happen to the different site. For the acetylation, the enzyme just conjugate such a lipid. It's kind of post-translational modification. When the protein, after protein synthesized, the enzyme in the cytosol or in the lumen of the, in the cytosol, they will conjugate or modify certain lipid to the N-terminal of the amino acid, okay? So this protein will contain the lipid on their polyamino acid. So acetylation will happen to the cytosolic leaf flap and in their conjugate to their N-terminal, and they will use this lipid to anchor to the cell membrane. Prenylation will happen to the cytosolic leaf flap, and also and then conjugate to the C-terminals. It depends on the enzyme. If they happen here, it means the enzyme to do this job exists in the cytosol. If the enzyme do conjugate with the build up the GPI anchor, it means the enzyme could be either in the extracellular space or usually in the Golgi lumen. Okay, it's because they need to do the modification in the Golgi lumen. Then such an orientation will kind of transfer, will keep such an orientation toward the plasma membrane. Then it will become the extracellular space. Okay, so the enzyme to do the GPI anchor will exist in the Golgi lumen. For example, the glycolysis always happens in the cytosol. Why? Because the enzyme required for the glycolysis expressed in the cytosol. Like GAPDH that is expressed in the cytosol. So why oxidation, phosphorylation only happens to the mitochondria? Why mitochondria contain that function? Because those enzymes required for oxidative phosphorylation only expressed in the mitochondria. Okay, yeah, that would be kind of principle. Okay, so the GPI anchors happens to the exoplasmic leaf left. It's kind of conjugate to the C terminals. And so that is why they happen to this cytosol part or to the extracellular space, because the enzyme to do this job, they express to the different side, okay? So I hope I just kind of, to explain this, then you kind of will gradually have more and more sense. When you see your result, you probably will guess what's the next step you should do. Okay, you need to find out the enzyme. Or if you do, for example, you find the modification of your protein, then you do the RNA-seq, you find a lot of candidates, but you just kind of, you don't need to pick, you don't know how to pick up the right one, right? You need to follow the logic. That would be the logic. Yeah, of course, if you follow the logic, you still can find kind of, and then you know what is the expected result, what is the surprising result. That would be the sense of the cell biology. Wow. Okay. Okay, and many surface protein, they are glycoprotein. It means you will conjugate a lot of sugar on those polypeptide. And usually, so the Golgi lumen contain a lot of enzyme. Here in the lumen, they will do such a glyco reaction. They will conjugate, mainly carbohydrate will conjugate to the serine surrounding and the asparagine. And then finally, when they go to the plasma membrane through the vesicle, they will arrange to the surface of the cell. Okay, and such a sugar or glycoprotein on the surface is really important for the cell-cell communication and many signaling outside the cells. Okay, for example, the human ABO blood glucose group antigen will be kind of regulate tightly through the glycoprotein. So you need to know the textbook only show kind of several glucose sugar here, but this is a red blood cell. They have a lot of membrane protein on the red blood cell, and those are membrane protein. They have a certain sugar polysaccharide here will be here. Okay, so those are conjugate with the surface membrane protein. You need to know where they are. Okay, so with this is a basic unit. Consider this is a basic unit. Okay, it's they will conjugate with such a different sugars. If you and we consider the people carry this will be the O blood type O blood. Okay, but if the people contain the gene can express GalNAc transferase, it will conjugate one more GalNAc, this sugar, to this basic unit. We define this as A antigen, and they will be the type A blood people. If the people, their gene, they have such a gene can express galactose transferase, they will conjugate one more galactose to this basic unit. It could be considered a B antigen. Okay, so they will be the type B blood people. If the people contain both this gene, they can conjugate this and galactose. They will be the AB, type AB kind of peoples. Okay, so in the bloodstream, this is blood vessel. Okay, in the blood there will be many blood cell, right? And the solution will be the serum. Okay, so we have A antigen, A antigen, or B antigen, or AB antigen. And in the serine here, serine is the solution part, okay? We have several antibody, okay? So for the A blood type of the people, they contain the A antigen on their red blood cell. But in the serine solution part, they have anti-B antibody, which means this antibody will bind to the B antigen, okay? Luckily, they don't have B antigen on their red blood cell, otherwise they will die, okay? And for the type B people, they will have B antigen, but anti-A serines. Or AB will be A and B, they don't have any kind of antibody. And the O blood cell, people don't have any kind of, they only have a really basic unit of their antigen, and they both have anti-A and anti-B antibody. So, so if the anti-B, when you donate your blood, it also contains kind of antibody in your serum, right? So if anti-B antibody meets red blood cell with a B antigen, they will conjugate, will bind to the B antigen, and form such a cluster. And it will form a lot of cluster in your body, and finally will damage your circulation, and you die. Okay, that is why they cannot receive if they have such an antigen, they cannot receive the blood from the A person, something like that. Okay. Okay, so that would be kind of example for the lipid anchor protein, and we'll also talk about the glycolipid on the surface, how they kind of affect our life. And next, this part we just introduce the polyphenol protein. So phospholipids, as I mentioned before, they're the ester and ester linkage, right? Ester will be weaker by ester because phospholipids, this will be the enzyme to damage the phospholipid. They will be easy to disrupt ester linkage, but cannot easily disrupt ester linkage. Okay, so this is a big family. It contains A1, A2, C or D phospholipids. They will damage. This is the polar head chemical structure of the polar head lipid. So it will damage this bond, this bond, this bond, this bond. If that lipid needs to be kind of destroyed and do the recycle or reuse, those phospholipid will do their job to disrupt those, break down the chemical bond and then destroy this lipid. So, but how the enzyme all need direct interaction with their substrate. There's no way to do the long distance remotely reaction. That would be the super surprising example. No way. Okay, enzyme need to direct interact with their substrate. So phospholipid to digest the phospholipids to digest the phospholipid, they also need to do the direct interaction. So this is the structure of the phospholipids A2. Okay, it will disrupt this bond. How can they kind of collect the lipid? First, inside, this is a cartoon of phospholipid 2. In the interior, this part, there will be the tunnel here. And the entrance of the tunnel, they will arrange a lot of polar amino acids. So we call that a polar ring. So those polar ring will bind to, this is lipid bilayer. I have a polar ring. It will have affinity with those polar head. And it kind of suck out a little bit those lipids, okay? And which lipid they should suck out? Because if this lipid, they have a certain damage, they will be easier to be sucked out because they don't have a beautiful or good affinity with other lipid. So they kind of more easy to be sucked out, okay? So they will bind here and suck out such phospholipid. Once they suck into the tunnel, the wall of the tunnel covered by many, many non-polar amino acids. So this fatty acid will be super happy aligned with those wall, okay? And they arrange a calcium ion on the end of the tunnel, which will kind of help them to bind with the polar head, okay? Then they have a chance, they have a direct interaction through their active site, catalytic site to disrupt this bond, okay? So that is why they can disrupt this. They use a calcium to bind those chemical, this is the tunnel. They use a calcium to bind this polar head and digest this bond, okay? So this is the way how polyphenol protein associate with the lipid and do their job, okay? That would be the example. Okay, and also the protein, when you do the experiment, you always, when you study the membrane protein, you always need to use detergent. Detergent are kind of chemicals, and they are all amphipathic. You can see here, the yellow will be the those are 4 typical detergent, and the yellow part will be the hydrophobic part, and the green part will be hydrophilic part. Okay, so but their structure, although they are amphipathic, their structure are not kind of similar to the phospholipid. That's super different. Okay, so when you incubate the cell with those amphipathic molecules, the hydrophilic part will align with the lipid hydrophilic polar head. Hydrophobic part will align with the kind of hydrofatty acid tail, but their structure and their structure are quite different, so they would destroy the original alignment of the lipid bilayer. That is why we damage the lipid. Okay, so it can go super fast. Okay, for example, this is a cell. You just add the detergent. This is the membrane. You will see add the detergent within 5 seconds. It can disrupt the membrane. That would be the way why we can kind of use a soap to wash our hand, can kind of clean the pathogen virus, because we damage their membrane. Okay, they cannot survive without the membrane. Okay, so all these kind of chemicals, detergent, they are amphipathic. They can disrupt membrane, so they can help you to extract the membrane protein. But they kind of have 2 different groups. One is ionic detergent. The other one is non-ionic detergent. For example, SDS belong to the ionic detergent because they have a charge. Okay, and the Triton X100 is non-ionic detergent. They do not have a charge in these groups. What's the manner to have charge or do not have charge? All of them can help you to extract the membrane protein, but the one with charge, once they extract the membrane protein, they will also bind to the protein based on their charge. They will kind of, if the protein need to be kind of functional, they need to properly form and the proper 3D structure. But binding with those strong chemicals, they will kind of make them become amino acid sequence, only just one chain, without kind of, we call that denature. They're still there, but they don't have activity because they are unfolding at all. Okay, but if you use a non-ionic detergent, you can kind of extract the membrane protein with minimal damage to the enzyme activity. Okay, so that is why if you don't care about the protein activity, you can do Western blot. You only check their expression level. You can use SDS in your SDS page, right? But if you want to measure the enzyme activity, do not use SDS, because you have no chance to keep their proper 3D structure. And if you can measure the enzyme activity, there will be surprising result. Okay, right? So if you want to measure the enzyme activity of your membrane protein, use a non-ionic detergent. If you don't care, and also want to kind of denature the protein, that is why people use SDS page to do that. Okay, and also people also care about one thing, is critical micelle concentration during the protein extraction. It's kind of stressful. If you above this level of the certain concentration of this critical micelle concentration, above that concentration, that means you put enough amount of the detergent and the protein can be retained native-like conformation. They can keep proper folding more likely. But if you don't give them enough amount of the lipids, sometimes you still can extract them, but it will kind of only align with them through this. So sometimes they will form the aggregation and it will trigger some precipitation, okay? And those proteins, sometimes they are partially unfolding, okay? So that would be the one things you need to care about. Okay, so just quickly summarize. First, we talk about the lipid anchor protein. We use a GPI anchor, acetylation, prenylation. Where do they happen, and which N terminal or C terminal that you need to remember. And the protein, we use a phospholipase to, as an example, to show you they use a polar ring to associate or suck out the lipid, and then they have a tunnel to digest those damaged phospholipids. And we also showed you the integrate or transmembrane protein. Usually the transmembrane domain, they are alpha helix and a non-polar amino acid, but they always have kind of exception. So the porin use a beta strand barrier as their transmembrane domain, although they still arrange the hydrophobic amino acid on the surface of the beta strand. And also you can find certain polar amino acid, for example, in the T cell receptors, which can use the electrostatic force to help them to stabilize such a protein complex. So we also talk about the most surface protein. They are glycoprotein, for example, the ABO blood groups. Okay, and the detergent, you will see the ionic detergent and the non-ionic detergent. They both can disrupt the membrane structure. But the ionic detergent, they will denature the protein. Protein is still there, but they don't fold anymore. But non-ionic detergent, they don't denature the protein. They still can allow you to measure the activity or characterize the enzyme activity of your protein. Okay. Any questions so far? Okay. Okay. So the next section, we will talk about the phospholipid synthesis in the ER membrane. So the lipids are mainly synthesized in the smooth ER. Okay, because there are several key protein required for the phospholipid synthesis only expressed in the ER. Okay, this will be this protein only expressed in the ER. Okay, so phospholipid synthesis is a multiple step process. In the beginning, their material is acetyl-CoA, so you can get acetyl-CoA is a super important intermediate metabolite. Okay, you can get that from for example, glycolysis or many other reactions. So in the cytosol, you can see the acetyl-CoA. They will use a certain cytosolic enzyme to become the fatty acid. Okay, and such a fatty acid will conjugate with the CoA, become the fatty acid CoA. Then those fatty acid CoA will diffuse in the cytosol. Okay, every 10 seconds they can move around the whole cell super fast. But once they approach to the plasma membrane, the two enzymes GPAT and LPAAT can transfer those kind of fatty acid CoA will conjugate with those glycerol phosphate and become phosphatidic acid and incorporate them into the ER membrane. Then the phosphatase will remove those phosphate group and become diacetylglycerol components. Then the choline phosphotransferase will transfer choline, conjugate them with those lipid and become the phosphatidylcholine. Then they originally express right after synthesis. They originally express to the cytosolic leaf lab, but there will be one flip pass determine their asymmetric. They will use energy to flip that to the exoplasmic leaf lab. Okay, so that is why the PC will accumulate to the exoplasmic leaf lab. That is why I claim the flip pass and the flop pass, they will be the guide to determine the asymmetric. Okay. But there will be the one issue. You will see the fatty acid, it's kind of lipid, it's kind of hydrophobic molecule. Such a hydrophobic molecule, how can they kind of feel comfortable in the cytosol? They should be hydrophobic, right? The cytosol is a hydrophilic environment. How can they kind of be delivered or through the cytosol properly? They need some help. Okay, the helper is, for example, the fatty acid binding protein. This is a soluble protein. Soluble protein always have a kind of surface, hydrophilic surface. So that is why you see a lot of beta strand. They are kind of hydrophilic on the surface, but they have a pocket, hydrophobic pocket in the interior. Okay, they use that pocket to collect the fatty acid tail. And one fatty acid, okay, so fatty acid will bind here and they kind of freely diffuse to the cytosol and help them to kind of properly kind of approach to the ER membrane. Okay, they don't stuck with other hydrophobic component. Okay, so fatty acid FABP binding protein can do this job. And next, we will kind of provide more detail about this lipid droplet formation. So as I mentioned before, if the ER synthesize too much lipid or cholesterol or triglyceride, they will kind of accumulate lipids to such an intermembrane space. Why they will accumulate lipid there? Okay, that will be the question we try to address. So researchers previously, they try to identify, because we like to eat, but we don't like become fat, right? So the one way is it possible we eat a lot but still cannot generate lipid? Our cell cannot synthesize lipid efficiently, okay? So they want to find out the key enzyme synthesize lipid. And if they can find out the key enzyme synthesize lipid, they can design the inhibitor. So you can, before you eat the food, you just eat those inhibitor. You can eat without synthesizing too much lipid. That would be the perfect situation, right? So that would be their motivation to find out the key enzyme for the lipid synthesis. So after such a screening, for example, they many conduct this experiment in the yeast or in the mammalian cell, I forgot. They just try to incubate the yeast cell with the kind of oil red, which is kind of lipids. Usually, after oil red incubation for a couple hours, you will start to see the lipid droplet. So they can kind of, the yeast can kind of, they can generate the random mutation or the library of the yeast. And they just find out which mutation can help yeast do not produce lipid droplet. And finally, after screening, they found DG81 and DG82 are the two proteins really important to kind of synthesize such a lipid droplet. And after identifying those are key enzymes, they keep doing, for example, their protein structure, why they can do this. And they found DG81, this is the protein structure of DG81. They have a cavity in the site. This will be their transmembrane domain. They have a cavity in this site. After kind of acetyl-CoA, the material of the triglyceride are acetyl-CoA. After the material go into this site and they will generate triglyceride and the cholesterol, they will kind of directly go out from this site. That's exactly the space between the two leaflets. That is why the lipid will accumulate here, okay? Because I generate lipid and just put the other side. They go out from it. That will be the exit side. And that is why the lipid accumulate here. And they are pretty comfortable there because this is a hydrophobic environment. They just stand there, okay? Next, how can such a lipid kind of finally budding out to form the lipid bilayer, lipid droplet? Will be the other protein called Seipin, S-E-I-P-I-N. So this is ER membrane, and Seipin is kind of transmembrane protein. So this is one Seipin, and this will be, from the top view, this will be oligomer of the Seipin, okay? We just kind of ignore transmembrane domain here. You will see here, there will be a lot of hydrophobic helix here. They kind of without the lipid accumulation, those Seipin, they just freely electrodiffuse in the ER membrane. They kind of evenly distribute in the ER membrane. But once DG81 synthesize a lot of lipid, they will bind with the lipid here through their hydrophobic helices, showing in the purple color. And those lipid will also help the Seipin to increase the local concentration. And finally, they will form the oligomer here. This is top view. So they kind of can synthesize and kind of positive feedback loop. They will accumulate more lipid and accumulate more Seipin. And finally, they will become a budding hot zone for the budding out. And finally, accumulate more and more lipid. And with other kind of protein helper, they can form the lipid droplet and budding out from the ER. Okay. For the cholesterol, this is the biosynthesis pathway of the cholesterol. And the HNG-CoA reductase, this reaction will be the key rate controlling step, okay? And HNG-CoA is the ER integrate membrane protein, which solely in charge in this key rate controlling step. If we accumulate too much cholesterol in our body, we will have a big trouble. For example, in the blood vessel, if you accumulate too much cholesterol, those blood vessel cells, they will become inflammation, and it will go to certain proliferation or reorganization. And finally, those blood vessel will become thicker and thicker. And then finally, this part physically will become really weak and a kind of disruption happens. So to kind of prevent the blood leak out, so you need to accumulate a lot of thrombus platelet here, okay? And they will form the larger and the larger thrombus. And finally, will block the bloodstream and trigger the heart attack. That will be the disease called atherosclerosis, okay? So the one way to efficiently treat atherosclerosis is try to reduce the cholesterol. And the drug you need to design will be, of course, target to the key rate controlling step or the key enzyme. So people found statin is the inhibitor can block the activity of Ch10&11 reductase. So that is why clinically many atherosclerosis patients, they have been treated with a statin, and it's kind of really helpful for those patients, okay? So for the lipid, they are hydrophobic. How can they deliver in the cytosol environment? There will be three mechanisms. The first, they will use a vesicle to deliver a lipid between, use a vesicle, they will budding out. For example, this lipid, they will budding out with a vesicle and use this as a vehicle to deliver, going to their destination. So vesicle will be the one way. And in some cases, some integrate protein, for example, this is one organelle and this will be the other organelle. They will direct contact with each other. And certain membrane protein will in charge for this membrane contact site. And they will kind of help to exchange molecules between these two organelles for the exchanging. And also several binding protein, they have a hydrophobic pocket. For example, fatty acid binding protein, they will also help them to deliver lipid in the cytosol, okay? So in this section, we talk about the fatty acid can be used to synthesize phospholipid by multiple steps. Okay, initial step will be the cytosol, and later step will be the ER. And because why this happened to the ER? Because the key enzyme for the PC synthesis only expressed in the ER membrane. Okay, and the fatty acid binding protein can bind to. They have a hydrophobic pocket and can bind to those fatty acid and transport them in the destination. Ch10&11 reductase is a rate controlling step of the cholesterol synthesis. And also, we introduced the three mechanisms of the lipid transport. One is vascular transport. The other is integrate membrane protein can facilitate the lipid movement between the two membranes that contact with each other. And also, we have a small protein. Inside a cell, a small protein, for example, the FABP. They have a pocket to collect the lipid and the transport lane, help them to deliver in the travel in the cytosol. Okay, any